I'm Olivia, and I'll be introducing some of the best practices in cell culture from pipetting to culturing native cells. Oh dear. Oh, it's doing it that way. Okay, I'm going to try this. Okay, so there are basically two different types of cells, primary cells and immortalized cells. So primary cells come directly from a donor, from like a mouse or a human. So you take a tissue sample and prepare those cells from that. Whereas immortalized cells, you can like buy from the store, like our favorite ATCC. Primary cells, once you culture them or anything, they're no longer considered primary cells. So they're kind of like a one-time use. So we normally use them in like immunology or to study in vivo function. Whereas immortalized cells, you can culture them indefinitely. So we can keep those cell stocks and repeat experiments. And this is really good for studying tumor cells or just kind of replicating experiments. So these are two different types of cells that we're going to talk about how to culture each of these today. So to get started, we're first going to talk about immortalized cells, since these are a little more common. Okay, so to begin, how to culture immortalized cells. So like I said, you can buy them from the store. Most commonly, you buy them from the website ATCC. And what they're going to do is they're going to send you a little sample of cells that's in this cryo vial, as in picture 1. So they ship them to you on dry ice. So it's going to be frozen. So the first thing you want to do is thaw the cells. Now, this procedure is very important to take care to because cells are very sensitive in a way. So when you warm them up, you want to warm them up quickly. So to do that, you do that in a 37-degree water bath. So you take the little sample of cells and you just swirl it around in a 37-degree water bath for like 1 to 2 minutes, maybe maximum 5, just to warm it up. And then you go ahead and get started. Because the media that these cells are stored in is kind of damaging to them. So you don't want to keep them in this cryo agent for very long. So that's why when you go ahead and get them thawed, you dilute them in nice, comfortable media, and you centrifuge them to get rid of any of the extra cryopreservation agent that they are originally shipped in. Once you do this, you can go ahead and put them in your culture vessel and culture them in media and allow them to grow. So this first part of thawing the cells is really straightforward. You just want to be careful not to keep them thawed in their cryopreservation agent for a very long time because they will get damaged and you'll lose some cells. But once you have them in their culture vessel, again, you want to culture them with cell media. So to look into this, there's two main types, which is DMEM and RPMI 1640. They're very similar, and generally you can actually use both for most cell lines, but they're a little bit different in their calcium, phosphate, and glucose content. And actually, DMEM is mostly used for adherent cells, and RPMI is mostly used for suspension cells, but these are not all case scenarios. You can use both for either actually, and it shows that most cells are tolerant to both. But you can find out which media is best for your cells through ATC. It'll have a recommendation for you when you purchase your cells. Once you have your media, you're going to want to supplement it with two things. One being fetal bovine serum, which is also known as fetal calf serum, so FCS, you'll see that as well. And this provides the proteins and growth factors that your cells need to grow. And then you'll also want to supplement your media with an antibiotic. Most commonly, we use 1% penicillin streptomycin, and this is so that the bacteria, the antibiotic targets both gram-positive and gram-negative bacteria. So these are common, but I actually found out through reading that you don't want to culture your cells with antibiotics for very long. Maybe just when you first get started, but after that you really shouldn't, because it'll hide underlying contamination that can affect your experiments, and it can generate antibiotic resistant bacteria strains. So you don't want that either. So it's really not recommended to culture cells with antibiotic long term. Okay, so now you're growing your cells and they run out of room to grow. They become what we call confluent, which means that they are covering most of the space in the flask, so they no longer have any room to grow. So what you're going to have to do is you're going to have to subculture or passage your cells, which means that you're going to remove them from one container and split them into 2 or something like that. So this is pretty straightforward. There's a couple of methods of removal of the cells from the container. So for adherent cells, they're going to stick to the bottom of your flask. So you have to find a way to get them off the bottom, basically. So there's 3 different ways to do that. One is an enzymatic dissociation, in which we commonly use trypsin EDTA. Trypsin is a protease that cleaves proteins that enable the cells to adhere to the vessel. So once you do that, then the cells will be floating in the media. You can pour them into a falcon tube shown here on the left, and you can centrifuge it down and get your cell pellet and split that pellet as many times as you want, basically. Another way is to do cell scraping. So this way, when you have your cells adhere to the vesicle, you use what's shown here on the right, a little squeegee, to mechanically release the cells from the surface. This can be pretty damaging to cells. So normally trypsin is more recommended, but some cells are sensitive to trypsin, so you have to do cell scraping. For us, we only use cell scraping for raw macrophages. They're the only ones we need that for. But also there is something called versine, which is an EDTA solution for gentle non-enzymatic cell dissociation reagent. So the proteins that bind cells to the surface of the plate require calcium 2 ions so that those integrins can maintain adhesion. So the EDTA basically just chelates those calcium ions and allows you to remove the cells from the surface without having to use a harsh enzymatic treatment or anything like that. So people usually use this for more sensitive cells. So maybe primary cells that are adherent, you might want to use something like this for, because those are typically a little more sensitive. But overall, for most case scenarios, people use trypsin. So we have that there. Now that we're talking about calcium ions, I wanted to bring a question to your attention that I learned about from the DINISCO lab this semester. We normally use PBS in our lab to wash our cells and do a variety of cell techniques. But I did not know you can buy PBS with either calcium and magnesium or without it. And it's recommended that you can use PBS without these ions for cell washing and just minimal procedures. But if you're going to want to leave the cells sitting in PBS for an extended period of time, you want to include calcium and magnesium in your PBS solution. This way that it maintains cell adherence. So for example, when you're staining your cells, you might leave them for 15 minutes or more in PBS. And if you don't have the calcium magnesium, your cells might become unadherent to the plate. So that's just an interesting tidbit I learned from the DINISCO lab and something we might want to consider for our lab moving forward. Okay, so I brought a few images of how to subculture cells to your attention. And here I included a video where people use trypsin to do this. So you can kind of get a little bit more of a visual of what's going on. Okay. So this is again for adherent cells where you have to remove them from the plate. So first what they're going to do is they're going to use a sterile pipette and remove the media from the cells. And they're going to discard this media into a waste container. Next, they're going to rinse these cells with PBS. Again, this time you wouldn't need calcium magnesium because you're only doing a quick rinse. And this is to get rid of any remnants of media or serum that could inhibit the trypsin from dissociating the cells. So they're just going to remove the PBS using a new pipette. You only want to use pipettes one time, so do not reuse them because that ruins the sterility of the instruments. And now they're going to add their trypsin solution to their cells. Now their trypsin contains phenol red, but you can buy it without it, and ours is clear, but it's the same thing. They're going to go ahead and add the trypsin. And then they're just going to make sure that the trypsin covers the entire surface and all of the cells. And then incubate it for 1 to 2 minutes, maybe 5, no more than 10 normally. And then you can check under a microscope to see if the cells are floating or if they're still stuck to the bottom of the plate. So that's it.
Yeah, I have a question. Sorry. So you mentioned the time of incubation should be maximum 10 minutes. What will happen after 10 minutes for cells?
Trypsin can sometimes be a little bit toxic. It is a protease. It's not specific to cleaving integrins or anything like that, so it'll start damaging the cells. I actually have a slide coming up about that where it shows the toxicity of trypsin.
Okay, thank you.
Yeah, so like Maria taught me like only 1 or 2 minutes. But from the study that I'll be showing you in a few slides, actually it might be the next slide.
Michael has a question that he typed into chat about when staining, would we not do calcium, magnesium after we and would it be adherent?
I'm sorry, you kind of broke up a bit. I was talking more about staining cells for imaging where you want to stick them on the glass slide and you don't want them to come off the glass slide. because then when you wash them after staining, they're not going to be adherent anymore and you're going to wash away all of your cells. I wasn't specifically talking about for flow because those are non-adherent cells anyways. Normally the spleen ones are suspension cells, so you don't have to worry about adherence. I don't know if that's what your question was.
Just to add on to that, having the calcium and magnesium in the staining doesn't affect it, especially in your case, even if you were to use raw cells for flow, because you would de-adhere them after you do all this.
That's a great point, Candace. Okay, so this is what I was going to talk about, Arzo. So this is kind of an overwhelming thing, but don't worry, I will break it down. So this one group wanted to test to see the effect of different enzymatic treatments on dissociating cells and the resulting viability of those cells. So here we have trypsin. At 10 minutes, you see about 85% of the cells are viable. It seems pretty fine. And here's 20 minutes. They seem fine. I personally, my experience, I don't think 10 minutes is plenty, but see, you can see 30 minutes, it kind of goes down to like 60%. It starts going down. So the longer you leave it, the more damage it's going to do, basically. But it's not super harsh. But what we can see here is they used a pipette and a pasture pipette to do like mechanical dissociation, pipetting up and down. And you can see it really does a lot of damage to the cell. They unfortunately didn't include a cell scraper, but from our experience as well, that can also cause a lot of damage to the cells. So that's definitely not the first choice. But what they did is they were trying to make single cell suspensions, particularly. So they included using enzymes in the addition to mechanical suspension afterwards to break up the pieces. And you get that it seems to be fine then. The mechanical use is fine once you actually dislodge them. And what they also did is they combined two different enzymes for dissociation. So particularly they were interested in checking DNase 1. And this is because when you have a clump of cells, it's because free DNA is causing them to stick together. So they include DNase 1 to help break up those clumps, and that gives you a better single cell suspension, and it seems to do pretty well with viability. And they also tested total cell count to see if like it was dislodging the cells from the plate fully. And you can see trypsin does an okay job for only 10 minutes, but mechanical dissociation is really hard to get them off. They're pretty sticky. With cell scraper definitely works better than pipetting though. Using enzymes and mechanical force is a little bit better, but they do recommend still using like a combination of trypsin and DNase, kind of, if you're really wanting to get a good single cell suspension. But normally you have a confluent plate, like you normally have plenty of cells. It's not a very important thing to get 100% of the cells back necessarily. Okay, so once you have your cells dislodged from the plate, you might want to count them. This is really important for experiments as well, to make sure you know how many cells you have, but it can also help you have a better estimate of how many cells to seed and to freeze and different things like that. So what you're going to want to use is the mechanical method here is a hemocytometer, which is shown up here in the top left corner. And highlighted in red is this tiny little grid. It's a little hard to see, so I went ahead and enlarged it for you down here. So basically what happens is you go ahead and add 10 microliters of your cell suspension onto here, and you'll see that each of these white corners will be covered in cells, so to say, which is shown here. And what you're going to want to do is you're going to count the number of cells in each of the 4 white squares. Okay, and you're going to include cells that lie on either the outer edges or the inner edges. So here, like this blue dot would maybe be counted if you're counting the outer edges, but these ones would not. You would only count 2 of the sides. Once you total up the number of the cells in all 4 of the quadrants, that would be your total cell count shown in this formula here. You would divide by the number of squares being 4 and multiply by 10 by the 4. And this will give your cell concentration in cells per milliliter. And some people use this little, it's like a little clicker. So when you're counting all these cells, it will help you. You don't have to count them, I don't know, in your head. You can just click the button and that'll help you count them a little better too. But moving into the future, there are things called automated cell counters. It's fantastic. There's a little glass slide and you put like a little 10 microliters of your cells on the glass slide and insert it into the cell counter. And within 10 seconds, it tells you your cell concentration. And it is just mind-blowing, really, what the future holds. And then also another little tidbit that can be helpful is you can add what's called tryphin blue to your sample. This helps give you a cell count of your live cells and not include dead cells. So tryphin blue stains your dead cells blue, so you can see this dead cell is darkened. And then also your live cells will show up more transparent. So that way when you're counting your cells, you can count just the live cells if you want and give you a more accurate cell count. Then I went ahead and included in here for you guys a number of cells per milliliter kind of concentration for different culture flasks. So here are different size flasks, if you want to do plates, if you want to do well plates, these are the recommended cell densities. And this can also kind of differentiate between cells a little bit, because if you think of splenocyte cells, they're really tiny compared to macrophages, which are much larger. So it'll vary a little bit, but this can give you a good starting concentration of things you want to look at. Okay, so talking about these flasks and plates, these are things to consider how you want to culture your cells and what you might need for an experiment.
Hey, Olivia.
Yep.
I had a question on the last slide.
Yep.
So from a lot of literature that I read, it seems like people tend to break that number of cells per, like in a 24 well plate, it says it recommends 1 × 10⁵. And I see a lot of people tend to do 1 × 10⁶. What effect would that have?
Okay. So again, the hard thing is these don't have very good generalizabilities because it really depends on your cell type. So some cell types like to be more confluent so that they have better cell signaling, where other cells might like to have room to grow. Like our 14-1 cells grow really quickly, and so it's better to keep them at a lower confluency compared to other cell lines. So it's very hard to generalize this, but also, like I've explained to you before, it also kind of depends on your experiments. These I would consider that you want to have them in the flask for like 2 days. Remember, and then if you're going to do the experiment the next day, you'll want to see them at a higher confluency so that you have enough cells for your experiment. So it's very dependent as well, but I found that this was a decent generalizability to go ahead and get started. Like I said, 1 × 10⁵ and 1 × 10⁶, I can't imagine there being that big of a difference. I think they'll both survive, you know? I think
well, I guess my curiosity comes because for macrophages at least, I don't think they're that closely packed. So it's interesting from papers I'm reading is they pack these in 1 × 10⁶ for macrophage, and I just feel like I don't know how, I don't think they're that densely packed in the body. So I guess I wonder if there's other associated effects that occur when you pack them that way. I don't know, it was just curiosity, I guess.
Yeah, I think they're pretty amenable to how many, like what the confluency is. And again, for me, it's more important on timing, like how long you want to keep them in that vessel depends on, really determines the confluency you want to start with for me. But yeah, I like my cells more confluent than other people. But yeah, I guess I don't know how that translates to, like you said, how it exists in the body. That's for sure. But it definitely is a different environment. So the confluency might be different there as well because the environment's different.
Yeah, thank you.
The same cell signaling and growth factors and stuff in this artificial environment. So maybe they need more friends. They're signaling. But that's a good point. Okay.
All right.
So for cell culture, there's 2 different common vesicles to use, or vessels, sorry. So we commonly use flasks to grow cells like long-term in our incubators. So they come in different sizes in this picture in the top left. And you can grow them there. They have a nice cap on there that has a filter to prevent any, kind of limit the contamination of stuff getting in and out. But some people prefer to use Petri dishes, shown in the bottom left. I think that these work really well personally for cells that you have to do cell scraping on. Because if you can imagine getting a cell scraper into the flask through the little cap is really hard compared to this open dish. I think that works really well. But the culture ones are definitely better to like reduce contamination and stuff like that. So that's what we mostly use. And also experimental, we use well plates and glass bottom dishes. These are just examples of things you can use experimentally. So if you want to do fixed cell imaging, you normally use a well plate with a glass cover slip shown over here, placed at the bottom. And then if you want to do live cell imaging, you can do a glass bottom dish, which basically has the glass cover slip built in. And then you can use the 96 well plates for MTT assays and all sorts of things. So these are normally the vessels you use if you're wanting to seed cells for an experiment. Just a little bit of an introduction to that. Okay, so now how to freeze cells. This process is very sensitive as well, just like the thawing. So you have to resuspend the cells in a cryopreservation media. So they don't just go in regular media and go into the freezer. And this is because the water will freeze and turn into ice crystals. So you normally add a cryopreservation agent such as DMSO to prevent that from happening so much and damaging the cells. So in our lab, we use 90% FBS and 10% DMSO. But I actually read online that you can use 90% just supplemented media and 10% DMSO as a cost-friendly alternative. So I think we might give that a try. People have said it works just as well. And to back that up, I found some literature here that shows the amount of DMSO to include. And in the bottom here, it shows that if you have FBS or FCS, which are synonyms, or 10% FCS, it doesn't really affect the cell viability after freezing the cells overnight and then thawing them again. So I think we can maybe lower the amount of FBS we use in our cryopreservation media. And also here in the top, they tested DMSO concentration. This is the cryopreservation agent, so that's what could be kind of toxic to cells. But they found that 2-3 molar seems pretty fine. It's 50%. You're still not going to get a lot of cells back necessarily from your stock, which is normal. Some people normally report 20% viability from frozen stocks, but these ones suggest around 50. 1.3 molar is 10%, so we're kind of in this range here is what we're currently using. So I guess we could increase that if we want, but it seems to be working okay for now. And so again, you're going to put this in a cryo vial, which has a recommended concentration of 1 × 10⁶ cells per milliliter. And you normally do it in a 1 ml vial, and this will give you enough cells for like your T75 flask for when you freeze them or thaw them. Okay, the most important thing though when you're freezing cells is to control the cooling rate. You do not want them to cool rapidly, so you want to control that to 1 degree Celsius per minute. And there's a couple of ways people choose to do this. One is the most expensive way possible, using a controlled rate freezer. So this basically, you can just set the rate and it'll freeze it at that rate. But I think these are really expensive, and I don't know why you would have one unless for another purpose necessarily. So mostly people use ethanol-based cryo chambers. So this is where you fill the bottom of the chamber with ethanol, and then put your little cryo vials inside of there, place them in the -80, and the ethanol slows the cooling. So it works a little bit better than just putting it straight in the -80 or in the liquid nitrogen. I also read online that there's a really good cost-friendly alternative where people recommend using styrofoam containers like the Falcon tube racks and just putting your cryo vials in there and putting one lid on top and one on the bottom and taping it shut. And you can put it in -80, and that'll also reduce the cooling rate. So I heard that works pretty well. But Arzo learned from the Chin Lab from Zhonghe that they just put theirs at 4 degrees Celsius for a few hours, then move it to -20, and then put it -80, and then finally liquid nitrogen. So they just do it that way, and it seems to work really well for them too. So I think it's just the more control you have over your cooling rate, it's going to help with your cell viability. But overall, as long as you get some cells back, it should be okay. And then, of course, for long-term storage, you're going to want to put them in the vapor phase of liquid nitrogen. So we have these little boxes in our lab where you can put a bunch of different cryo vials in there, and then you can see here in the liquid nitrogen container, you can store like 10 boxes. So our friends in the Chin Lab have this and allow us to store our cells there for long term, and that's really where you want to store your cells. Okay, so most of what I went over was about adherent cells or immortalized cells. So I just wanted to touch a couple of different points about if you're doing suspension or primary cells. If you have primary cells, you're not going to thaw them because they're coming straight from a donor, so you don't have to worry about that. Passaging for suspension cells, you just don't have to worry about the dissociation step. You can just pour them straight into a Falcon tube, centrifuge them down, and resuspend them in your media. You don't have to worry about trypsin or anything like that. And for freezing, is not recommended for primary cells, again, because once you culture them one time, they're no longer considered primary cells, and they're much more sensitive, so they do not do well with cryopreservation agents. Okay, so primary cells, as we mentioned, they come from a donor tissue sample, so you can't just really buy them at the store. So this is how you can make a single cell suspension from a tissue sample. So you take a surgically resected sample, such as like a spleen from a mouse, and you store it in PBS, and then you bring it upstairs. And these people went ahead and centrifuged it down to get rid of the PBS and resuspend it in a nice friendly media. Then once they do that, they go ahead and do what's called mincing the tissue. And people do this by pulling the tissue apart with either a scalpel, tweezers, whatever you can think of, just like anything to kind of break it up. And you can see in this picture here, now we have big, like smaller chunks of tissue, basically, in nice, friendly media. Then you can go ahead and centrifuge that down and resuspend it in, this is an enzymatic dissociation. So they go ahead and add collagenase and DNase or anything to kind of break it down even further into a single cell suspension. And then what you want to do is put this through a cell strainer, which collects the large chunks of tissue that didn't get broken down or like fatty tissue that's left over. So that way you have a nice solution of cells in here without any more chunks. So that's what the cell strainer is for. And then you can centrifuge this down and you'll get a nice pellet here. And then you can do red blood cell lysis if you want to collect just your white blood cells. But that's an optional step because sometimes people want the blood cells, I guess. But this is a lot of information to take in, so I went ahead and added a video to make this a little more clear. All this lady talking. Okay, so how to prepare a single cell suspension from tissue. So they have their little tissue sample sitting in PBS. They just kept it in PBS for this step. They didn't change it into media. I don't think it matters that much. Now they're mincing the tissue and they're just using the end of a syringe. So really whatever you can just to kind of break up the tissue a little bit more. Then they go ahead and apply their cell strainer and they're going to pour their minced tissue sample into the cell strainer to get rid of any remaining chunks. And then they're going to do like a mortar and pestle style method of breaking those chunks up a little bit more in the cell strainer, and then wash the cell strainer with PBS just to wash any cells that are stuck in the strainer. And then you can discard any of the leftover chunks of tissue. So now you have your cell suspension. So you can go ahead and centrifuge that so you can get your pellet of cells, which it'll show you how nice it looks here. So you go ahead and remove the supernatant and you can see the pellet there and it has red cells on there and those are your red blood cells. So you can go ahead and lyse those if you want to get rid of them and then your pellet should be white afterwards. But they do not show that step here. That's an additional step if you want to do that. Okay, next one. So that's how you do a single cell suspension. Okay. So now that you have your single cell suspension, you might want to isolate a certain type of cell that you want to study, like a T cell or B cells if you want to remove them. So to do that, there are 7 different methods that are commonly employed to separate or isolate cells. So the first that I'm going to talk about is immunomagnetic cell separation. So there is both a positive and negative form of this cell separation. So for the positive form, you take your original sample and you add a magnetic labeling kit from antibodies and stain this or label the cells that you want to keep. So you could get a antibody cocktail with magnetic properties that stain or bind T cells. Then you let that incubate for 20 minutes. Then you apply a magnet and you pour out the supernatant that contains all the unwanted cells and you have just the cells that you want at the end. And like I said, there's also a negative cell separation in which you get an antibody cocktail for all the cells you don't want, and then those are left and the supernatant contains the cells of interest. So you can do both a positive and negative form of immunomagnetic cell separation. So this is one way that you can isolate different cell types. You can also do flow cytometry kind of stuff. So what you do here is you again label your cells with antibodies, fluorescent antibodies, and then it passes through an electrical charge ring that assigns charge to each of those cells, and deflector plates allow you to separate those into separate collection tubes. And this has been shown to be able to separate really rare populations of cells. So for example, here, this cell type was only 0.2% of the overall initial cell population, and they got it to 98% purity. So this is really useful for rare cell types. They also use density gradient centrifugation and sedimentation, which is kind of a process we're familiar with through protein purification. So you can see over here on the left, all the different cell types have different densities, so they'll come out at different sedimentation rates. And you can also use a medium that you can choose what density you want it to be at, so it helps separate your cells of interest in a way. So this is kind of how the cells settle out. But you'll notice the red blood cells and granulocytes are kind of close together. So I found this cool little thing called hedacept, and it binds all the red blood cells together so that they come out more in like a pellet, like they sediment faster. So that way it's easier to separate your red blood cells and your granulocytes, and you don't have any little red blood cells like floating up into the granulocytes that didn't get pelleted out all the way. So this like really improves the purity of getting granulocytes. Okay, there's also adhesion, which is something I think our lab has recently started to look into, thanks to Arzo. So basically what you do here is you can take your cell, single cell suspension, put it on a plate. Some cells are going to adhere to the plate and some are going to stay in suspension based on their cell type. So if you take a set of monocytes or a whole leukocyte sample, which is all your white blood cells, the monocytes will adhere to the bottom and you can get rid of everything else, or you can keep everything else and get rid of your monocytes. And this can also be used for tumor cells. So you can see that the primary aggregates are in suspension, but there's also adherent cells. So you can kind of separate cells this way as well. And this is, I think Arzo used this to separate dendritic cells from macrophages, because macrophages are adherent and dendritic cells are suspension. So that's another way to separate those guys out. Okay, there's also immunodensity cell isolation. So how this works is you actually cross-link the red blood cells with unwanted cells using this thing called rosette sept. And that way it all comes out at the bottom, all the unwanted cells, and you're left with highly purified cells in your supernatant. So it works like this. You can kind of see the red blood cells are being cross-linked with unwanted cells with this rosette sept cocktail type thing. It's the same thing with the immune density stuff. Okay, and lastly, there's microfluidic cell sorting, which is the lab on a chip kind of stuff. So there's both active and passive. So passive is where you apply your cell sample and it'll separate the cells based on like size or shape. But there's also active where they apply like an electromagnetic field and separate them that way. So you can do that on the lab on a chip for cell sorting as well. Okay, so I found one review of these. A couple of these techniques were included to see which one was best for getting highly purified macrophages. Monocyte-derived macrophages. So first they went ahead and took cell culture, it was called a Buffy coat, which is like all the white blood cells, basically, and the platelets. So initially here, their flow cytometry just shows this, and then they separated they gated for the leukocytes, which is all your white blood cells. And then they were able to show that how many of them are monocytes, because remember before, the monocytes adhered. So that's how they separated them by plastic adhesion. And then they did both the negative and positive selection of the immunomagnetic separation, where they labeled the monocytes with the positive selection and labeled everything else with the negative selection. And you can see the plastic adhesion only got like 42% purity, where negative selection was 85, and the positive selection by immunomagnetic separation gave you a 99% purity. So it was the best method for isolating monocytes from Buffy coat human blood samples. They took this a step further and they incubated the monocytes with several growth factors to differentiate them into macrophages and so you can see they gated their leukocytes again and then separated their macrophages from their lymphocytes. And you can see it goes from 75 to 94 to 98.5% purity. So again, the immunomagnetic positive separation seems to give you the best purity, but the plastic adhesion doesn't seem bad. And from what I've read, you can also do several cycles of this. So you can remove the supernatant, leave it for a few more days, remove the supernatant to get rid of any of those lymphocytes and maintain your adherent cells and get higher purity of that. Okay, so as I mentioned, growth factors can be used to help differentiate cells as well. I briefly mentioned that on the last slide, but they are also used commonly to grow primary cells because they require a lot of growth factors to regulate their proliferation and growth and differentiation. So growth factors act as what's shown as the ligand here. So they bind to receptors and are responsible for a lot of signaling pathways basically in the cell. So they play a really large role. And one in specific I wanted to highlight was interleukin-2. So one of our colleagues at UTD recommended that if we wanted to grow T cells, that we should include IL-2 as a growth factor to help with their proliferation. So in this study, they had purified CD8 positive T cells in which they grew with no IL-2 for 21 days. And you can see the proliferation rate is like non-existent. But as you increase the amount of IL-2 growth factor that was included in the medium, it really helps with their proliferation. So if you're wanting to grow T cells, this seems like a really important growth factor to include. But there's a lot of growth factors. There's a lot of things to consider when you're growing primary cells, and it can be a little bit confusing. So I found
Yeah.
really awesome.
In your last slide with the interleukin-2, were they also adding any antigens to it as well?
No, they grew them with what are they? Feeder cells is what they call them, which is kind of like a mixture of other cells releasing other cytokines like macrophages and stuff like that.
Because I asked because I know that when T cells are when an antigen hits the T cell receptor, that's usually when the T cells will start expressing IL-2, and so the IL-2 will self-activate the T cell to proliferate. So they actually activate themselves after attaching to an antigen. So I know in our case, I don't think we would need to add interleukin-2, but I guess I was just curious if there was a study that also added IL-2 to and antigen to a T cell growth plate.
Yeah, I don't think they did that here, but yeah, we could definitely look into that. And I think, like I said, this is 21 days they grew these cells in culture. So I know ours, we don't normally keep them that long. Because you can see at 7 days, the IL-2 does not make that much of a difference, and that's like a week.
Right.
But if you're wanting to like grow and have a lot of CD8 T cells to work with, it seems like you might want to use IL-2 to help proliferate them.
And they said that this is for CD8, not CD4?
No, it's CD8 positive.
Okay, interesting. Okay. Thank you.
No problem. So I found this nice little poster that I want to send out to you guys because I think it'll be really helpful because I know for me the growth factors and cytokines, it just is kind of a mess. So this mentions all the different growth factors and cytokines that are important for differentiation of cells. So just to make this a little bit bigger for you, like for example, CD8, CD4 T cells, it shows the different interleukins that are needed to make T helper 1 cells, T helper 2 cells, T regulatory cells. So I just thought that this might be a really good reference for us to like post in the lab or something like that. It also includes monocytes and NK cells and B cells on this poster. So I think that just might be a good reference. Okay, dun, dun, dun. Contamination. So this is the worst part about cell culture. If you're not really careful, you get contamination in your cells and it basically ruins your life. So on the left, I have images of what bacteria looks like in cell culture. On the top, there's pictures of yeast, and here is virus, but you can't see virus. So you see over the course of 8, 12, and 24 hours, the cells like start dying because the virus is affecting them. So these are some things to look for in your cell culture if you are worried about possibly having contamination. And I included this video of what bacteria looks like because it's super depressing and upsetting. So they move around. You can see that in the video. There's little squiggly things that move around in your cells. So you can zoom in really closely on your cell culture to check for this contamination. Bacteria is like the only one you can really see by eye. I guess you probably could with yeast too. I haven't seen that in person yet, but this is what we are dealing with. But I have looked into it to see how can we avoid eating bacteria and is there a cure? So mycoplasma is another type of contamination, and it's really unfortunate because actually you can't really see it by a microscope, so it's hard to detect. But what happened here is this group tested a whole bunch of antibiotic type drugs to see if they could cure mycoplasma. So they took 100 different human and animal cell lines and incubated them with each of the recommended antibiotics at their recommended concentration, and they treated them for 14 days and then waited 4 months to see if the culture was either cured, if the mycoplasma came back, or if the culture died. So we have used sparfolaxin recently to see if this helps with some contamination problems. And you can see it only cured 33% of sparfolaxin. And then 62% it came back. So there's not really a cure. The best thing to do is just go ahead and throw that flask of cells away so it doesn't contaminate anything else and start fresh. But also ATCC, our friends for all references, is that they say that you can cure bacteria with penicillin, streptomycin, neomycin, and bacteriocin. So this is their antibiotic cocktail for curing bacteria. So I don't know, we can try that. But again, it's not recommended to culture cells with antibiotics for a very extended period of time. And this is because it can retard cell growth. So these are cells that were grown with either no antibiotic, gentamicin or pen strep, and you can see that it can really affect cell growth. And this is just for a week or week and a half. So imagine culturing them a long time. It can really affect the cell growth rate. So it's not recommended for long term. So the best idea for curing bacteria is to use what's called aseptic technique, or these are some good sterile technique suggestions from Thermo Fisher, how to be sterile. Okay, so when you get started, first thing you want to do is spray down the cell hood with 70% ethanol and give it a nice wipe down with a paper towel. This way you provide yourself a nice sterile surface to work on to get started. You also want to be wearing a lab coat and gloves and your hair tied back and all the PPE. You also want to spray down all the equipment and media bottles you will be using with ethanol and wipe them down. Make sure they're all sterile before you enter them into the cell hood. Now, this that they recommend for working in the cell hood. They recommend having your pipette on the right-hand side, your media bottles in the back, and your tubes on the left. And this is because a large source of contamination comes from crossing your hands over. So if you have like an open media bottle and you cross like the pipette over, you risk contaminating the bottle. So they try to keep everything separate and in an orderly fashion. So that way there's no crossing over and you're not like cross-contaminating kind of your media bottles and stuff like that. Image for okay. So next thing they're just going to show you want to use like sterile pipettes. So they come individually wrapped and you only want to use them one time. If you reuse pipettes, you're going to contaminate tons of stuff. So that's why they're like always throw it away like right after your use. And when you pipette out of bottles, make sure not to touch the edges or anything like that with the pipette tip. And something that I learned from the Danisco lab and from these videos and stuff is that you don't want to leave the lid off of any bottles. Like close them as soon as you can, like only have the lid off for a minimal amount of time. And if you have to take the lid off, they recommend setting it face down so that the inside of the bottle doesn't get like contamination from the air that you then like put on top and it goes into the solution. But I've also read that you should keep the lids on their side as well. But basically, you just don't want to set it down if you can. So again, don't reuse pipettes. And I think that's basically it from this video. So those are some really main things. No crossing over. Try to keep the lids on things. Don't reuse pipettes and spray everything down. And actually, at the end of this video, you spray down the hood as well when you're done. So once you're done, you go ahead and take everything out. They do not really store anything in their hood because it disrupts the airflow and limits the sterilization of the hood. And then again, when you're done, spray everything down with 70% ethanol and wipe it down. And that's the end of the video. Last thing, I want to leave you with a couple of tips and tricks. I read tons of ResearchGate forums on how to prevent bacteria contamination, how to maintain sterility, and what I found is summarized in the above. So sources of contamination. Incubator and hood, but water baths is supposedly the reason for 99% of contamination. So what most people recommend is replacing your water baths with magnetic beads, or just getting rid of your water baths entirely and warming your media in the incubator itself. So that is what has been recommended. There was a professor on there who trained undergrads in cell culture, and he would have 30 undergrads every semester who knew nothing about cell culture. And he said he always had contamination, but once he threw away his water baths, he has not had contamination for the past 9 years. So he swears that that's where it comes from. So that's definitely, I think, the first source to look for. Some people also said cardboard boxes are a breeding ground for bacteria, so not to keep them in the cell room. Technique, again, don't cross over open bottles, and they also do not recommend culturing animal cells and human cells together, because when you take animal tissue, it's not sterile and you're bringing it into the cell hood, so they don't recommend that. Supplies, don't reuse pipettes again. People, they recommend using a lab coat that is only in the cell hood or cell room. So for us, that's really important because we grow bacteria and we wear that cell or that lab coat and go into the cell room. And that contains all like the bacteria stuff. So they really recommend having a lab coat that's kept in the cell room. They also said no talking in there because then you're gonna spit into the cell hood or something like that. And to reduce the traffic in cell room as well, just kind of basically bring everything down to a minimum. But again, all of these are little tips, but everybody swears that it always comes from the water bath. So I think that's the main source. But you can prevent this hopefully by including a UV lamp in the hood to kill off bacteria. Some people even have a UV lamp in their room. Other people said try clean the water baths 2 times a week to reduce that contamination. And they also said you can do auger plate testing in the hood and incubator. So you just take an auger plate and set it in there, and that way any bacteria will grow on that plate and indicate where your contamination is coming from. The technique, again, spray everything with either 70% ethanol or 10% bleach spray. Keep your lids closed or set the lids on their side or face down to prevent contaminating there. Do not overcrowd the cell hood. That's why they remove everything and bring it in when they need to use it. Work with one cell line at a time to reduce like cross-contamination, and to aliquot reagents. So when you have those big media bottles, it's nice to pour them into a falcon tube. So that way, if you contaminate within the falcon tube, you're not contaminating the whole bottle and not sharing that with people. And then always autoclave your supplies that are not brought from a sterile environment. So any pipette tips or things like that you're using, make sure you autoclave them unless they're like individually wrapped. And they also really recommend washing your hands up to your elbows really well before any time going in the cellar hood to reduce any like dead skin cells from falling off and like stuff like that. But again, all minor. The water baths, people, that's where it's coming from. So I think we should try not dealing with those. Okay, that is the end of my presentation. I hope that it was a little bit informative.
Wow, great. Good job, Olivia. Can we get rid of the water bath? Is that feasible?
Yep. They recommend putting your stuff straight in the incubator because it's actually not a sterile environment because you open it to like the regular air and stuff. So they said just throw it in there and it'll be fine. They also said that you can like just leave it in the hood and let it warm to room temperature as well, you know? Other stuff like that. But they really said just get rid of them. There's no need for them. And when you like thaw cells, you can just get like a little beaker of more water and do it that way. Like you don't need the water.
I was gonna say that because I stopped using the water bath a long time ago because there's always something floating in it.
Yeah. Which my other concern is maybe the water baths inside the incubators too and stuff, but those ones we would just have to clean out like 2 times a week or something. They also said like you do, like you can add bleach and other stuff to it, you know?
We can't add bleach to the water bath because it'll start to etch the metal. It'll start to rust.
Yeah, they added some stuff too. I don't know if it was exactly bleach, like other sterilization stuff. But you can't put like ethanol because it'll create other fumes and stuff.
Okay. Well, that's great. Well, then I think we probably should get like lab coats for the cell room for people who do the cell room stuff. Because that's an interesting, I guess, it's people run from bacteria culture to mammalian cell culture. And yeah, I guess getting rid of the water bath would make a lot of sense. It is kind of scuzzy sometimes. And
Yeah.
all right. Michael, you have a question?
I had a question, but I kind of forgot what it was what it was about.
Okay.
I mean, maybe if you move back to your last slide.
Tips and tricks one or? Or the mycoplasma?
Oh, yeah, I mean, I had a question about the mycoplasma too. I know it wasn't about the mycoplasma, but so I guess in this experiment they only added plasma cure once, right?
Yeah, I think so. So it was a little bit difficult for me to remember. So they followed the recommendations of the antibiotic. So sometimes it was like they added 3 times 3 days a week or something like that. You know, like it was based on the recommendation. So I would have to look back at what the specific one was for plasma cure. It was, they did the treatment based on the recommendation of like the manufacturer. So they weren't all the same.
Yeah, I guess I see that it basically seems to cure about 91%, and it only has like a 6% culture death. So I just don't understand maybe why don't they do like a second addition to see if that further removes it completely.
You probably could for sure. But I can't remember. I saw something pop up. Did Candace say that plasma cure is really expensive?
It is pretty pricey. It's like $300 or $400 for one vial. And how they sell it. So plasma cure is designed to be like a one time, I know I have mycoplasma, this is how I'm going to get rid of most of it. And then you follow up with a plasmosin treatment. And I think that's like a week worth of treatment. So you have to buy, usually you have to buy both. So it's like you get the one to cure it, and then you try to treat to make sure that the mycoplasma is not there.
Yeah.
And I guess for the lab coats, I mean, you're walking with like bacteria all the time. I think the best you can do is just use a new lab coat each time. But no matter what, you yourself just have so much bacteria on you. So you just got to be as sterile as possible.
Yeah. Which I'm interested to see. Like I said, there's tons of tiny things, but it was really interesting to read that forum where they were like, you can literally not do any of the other stuff and just remove the water baths and you'll be fine. Like they really were like, it's not a big deal. Because if you see a lot of people who do cell culture don't even wear a lab coat, like a lot of times.
Oh, they do it outside their cell hood, like you mentioned.
Yeah, like the Danisco lab didn't even do it in a cell hood. They just did it on the counter, you know?
Yeah.
Mammalian cell culture?
Yeah. They did all their stuff on the cell counter. Like they try not to, but whenever they did the infections and stuff too, they don't, it's not a big deal for them to not.
But I'm pretty sure they sterilize the area before using it.
The desk?
Yeah, like their desk. I'm pretty sure they probably
Yeah.
sterilize the area. Because I mean, that's all we can do when we work with the mice as well. When we do anything with surgery, we just gotta use as best as we can aseptic techniques, but you can't control everything.
To be clear, the mammalian cancer cells and macrophages are BSL2. They have to, even if it's not a risk in contamination, you have to do it in the hood to protect yourself from them. I mean, it's very unlikely they're going to do anything, right? But they're BSL2. So when you manipulate them, you have to manipulate them in a safety cabinet.
I think the Danisco lab was using kidney cells. I don't know if they were cancers or not.
Yeah, the hamster kidney. Oh, those are just, what are those hex cells or something?
Mhm.
Okay. Okay, great. Well, if there are no further questions, let's all put our clothes on and turn on our cameras and thank Olivia for a really great talk. Yay! Very good. Wow, that's very nice. All right. And say hi to Al. Oh, I guess we'd say bonjour to Al Shaba now because he's back in France, right? Hello. Well, I wonder if we can get you back at some point, you know, when there's not a pandemic.
Maybe next year during my second year of master, I have a whole semester to do an internship, but usually they advise us to do it in France because after your internship, they can offer you like a job or something. So yeah, I'm not sure yet. I will see.
Okay. Sounds good. Okay. Well, thanks a lot, everybody. And I will, what is today? Monday?
Wednesday.
Oh, yeah, it's Wednesday. Do we want to do something on Friday with a Scrabble?
I am down.
Okay. Then on Monday, 5 o'clock, there will be a WebEx plus, or rather maybe a Zoom plus Pictionary game. Everybody can do.
Friday, not Monday.
You know what? It's Friday. I don't really know what's going on at all.
Friday
Where I am right now.
Friday is always scribble and drinking.
Yes. You don't even have to drink. You could just play Scribble. It's fun.
It's mandatory to drink.
No, it is not. Be clear, mandatory to drink. But you can if you would like. You're adults and it's 5 o'clock. Well, not you, one of you 20-year-olds, but 21-year-olds, adults. Okay. Well, I'll see some of you on Friday then.